Assalamu alaikum everyone. My name is Awais. I work as a postdoc in Loudia University of Technology. I'm in automatic control in the Division of Signal and Systems, Department of Computer Science, Electrical and Space Engineering. First, I would like to begin by thanking the organizer for a kind invitation to speak today. And I also sincerely apologize for not being able to join you live due to some unavoidable commitments. I'm sharing this presentation uh, as a recorded talk. Today, I will be presenting my research on using ultrasound for plant health and quality monitoring. Before we formally start the presentation, I would like to introduce my university and the research group that I'm currently working with. So let me start with a brief introduction to Luleå University of Technology. We are Sweden's northmost university with strong roots in industry collaboration and applied research. We have 4 campuses in Luleå, Kiruna, Piteå, and Skellefteå. Each is specialized in field ranging from space technologies to wood science to audio engineering. I'm located in Luleå campus. At LQ, we have a very strong focus on technological innovation. So we are working in AI, data science, acoustics, robotics, and simulation, just to name a few. The university also have extensive labs, and we have the highest percentage of external funded research in Sweden. So it makes it an ideal environment for experimental work like the project I'm going to present today. I'm part of computer science, electrical and space engineering, often known as SRT. And we have around 220 staff members, so 110 researchers, 70 PhD students, plus some administrative and technical support staff. We work with space technology and space systems, embedded and intelligent systems, signal systems, and computer science and digital services. So we also offer courses in master level programs and bachelor's level programs in the computing domain. We have labs for robotics, electronics, microelectronics, high performance computing. When it comes to machine learning and AI, we also have GPU clusters, HPC2N. You can search for this and you will have more information regarding this. And we have a strong applied and experimental research profile here in our department. I'm currently involved with the automatic control group. Here you can see the members of our team, spanning from professors and head of subjects to head of pavilions, to postdocs and PhD students. I didn't include the master thesis students in the slide. Our group focuses on control engineering, system modeling, signal processing, AI, and we are working with different projects, which I'm going to show in the next few slides. The projects we are currently focusing on addresses the global challenges aligned with the United Nations Sustainability Development Goals, whether it is clean energy, sustainable mining, resource-efficient food production, or digitalization in agriculture. Our research aims to make a meaningful impact on the society. In the next few slides, I'm going to present some work that we are currently doing. We are also actively collaborating with 𝗪𝗀𝗪𝗀, CISC in Spain, few other universities in Finland, even in UK, and even Brunei. And we are also very strongly collaborating with RISE, which is Research Institution of Sweden. I'm going to very briefly talk about ongoing projects. One important area of our research is automated model discovery for digital twins. We develop methods to automatically detect model deficiencies, perform adaptive updates, and maintain lifecycle management for the industrial digital twins. Another active project that we are currently working on is related to sustainable mining. We are trying to develop real-time optimization strategies for froth development processes within next generation digital mining ecosystems. Through this project, we are trying to develop practical methods for real-time monitoring and control within the complex industrial processes. We are also working with clean energy. We are currently studying some large-scale energy and material flows in fossil-free sponge iron production. The system must adapt to variability in renewable energy availability. Our role is to develop optimization strategies and robust control algorithms for this particular application. We are also involved in a very interesting project that requires intelligent pooling of data centers. And what we are trying to achieve is to reduce the energy consumption while maintaining the thermal stability. What we are currently doing is that we are doing some model-based and predictive control methods to reduce the energy consumption, and we are trying to maintain thermal stability as much as we can. So currently we are involved in food production projects as well. So currently we have 3 active food production projects. One I am going to present today, and other one which is basically a very big project, but I'm just going to tell you just an overview of it. So we are working on dynamic modeling and optimization of microalgae growth in bioreactors. The project aims to produce nutrient-rich biomass using sustainable processes, and the other one is the one that I'm currently working with, and which I'm going to present in the next few slides. With that background, let's focus on core topic of today, which is contactless wireless sensing for plant water status and early pest detection. We are very early in the project right now. It's been only 6-7 months that we are working on it. So the pest detection part is still not completed yet. We have efficiently detected the plant who was the water status in the potato plants, but we are working towards the pest detection. But detection of water status is directly relevant to the early pest detection as well. So let me just tell you what we have done so far. So from this point onwards, I will focus especially on how we use ultrasound as a tool for studying plants' health and quality. Ultrasound is a fascinating technology because it allows us to characterize materials non-destructively. In agriculture and plant sciences, most of the techniques that we are still using are invasive or destructive. Leaves are cut, samples are dried, or there are some biochemical tests that are performed. These methods, I'm not saying are not valuable, but they don't support continuous monitoring or automation. So using ultrasound is safe, fast, and repeatable. Unlike X-rays or destructive sampling, it doesn't damage the material. It poses no biological or environmental risk. In this project, our expected outcomes include developing characteristic acoustic map for plant tissues, proposing ultrasound testing setups optimized for agricultural environment, and establishing guidelines that enable farmers, researchers, and automated systems to detect plant stress, including early pest activities before the visual symptoms appear. So ultrasound in plants. So at first, the idea of using ultrasound on plants seems unusual. Most of the people associate ultrasound with pregnancy scans or industrial non-destructive testing in metals and plastic, but the fundamental principle is similar and simple, sorry. So ultrasound waves are just high-frequency sound waves, and sound can travel through any material that can vibrate. Plant, of course, they are also biological tissues. So leaves, stems, fruit, tubers, all of which are capable of transmitting ultrasonic waves. In the tissues, the waves interact with water content, cell walls, air pockets, and many other microstructures. And this interaction of ultrasonic wave can provide us very valuable information. As the hydration of leaf or plant decreases, the cells start to lose its trigger pressure, air-water interfaces change, and the mechanical properties of the plant shifts. Ultrasound is extremely sensitive to exactly these type of structural changes, often long before they are visible to human eye. So using ultrasound with plants can effectively become a communicator of their psychological state, and somehow we can say that we can listen to the plants using ultrasound. So just a little bit of context, what exactly is ultrasound? So ultrasound usually refers to waves above 20 kilohertz, which is beyond human hearing. In our work, we are using frequencies in hundreds of kilohertz range, particularly from 250 to 900 kilohertz. When these waves travel through plant materials, several key acoustic parameters come into play. For example, the frequency determines how wave interacts with different tissue structures. Wavelength affects sensitivity to small features. Velocity, the speed of sound in leaf, changes as they become drier or softer, and attenuation increases as the water decreases. So what make actually ultrasound is safe for plants, as I've mentioned before, unlike X-rays, it doesn't ionize tissue or alter the sample. It is also non-destructive, meaning we can probe the same leaf many times. And importantly, ultrasound can work in real time. A single measurement can take microsecond, which means the system can be integrated directly into automated platforms, even with robots, some IoT devices for continuous monitoring. So let's talk about the measurement electronics. Currently, we are using air-coupled broadband transducers, which are custom made. We didn't made it here in Lulio. We are working with a Spanish collaborator in Spanish Research Council. They can work with 250 to 900 kilohertz range. We have a high voltage pulsar from Ultratech, and then we have Python-based signal processing, and it is fully non-contact, so you don't need to touch even the leaves. So rather than showing you this diagram, which you can see on the slide, I can show you the electronics that we are currently using. So these are the transducers that we are using. Unfortunately, I don't have cables right now. I broke one of the cables, so I cannot show you the live demo. But here, like this, we set it up in a casing, and we usually put the leaf like this in between these transducers. I don't know if my camera can show it properly, like this. It's winter, it's snow outside, so I cannot find a very good leaf to show you guys, but here you can see the setup is placed like this, and then we measure using this pulsar. We use oscilloscopes as well, but this is something that we bought new, and the software have basically a built-in oscilloscope, and we record the signals, we record the different parameters that I'm going to show you in the next few slides. And using this, we measure the hydration in the leaves. Again, the setup is completely non-contact. We just have to place leaves in between these two transducers, and we are able to get some good results using this. When we do our experiments, we record different features. One of the most simple and powerful feature that we usually record is the peak-to-peak voltage amplitudes. You're going to see this abbreviation TLP, which is to-go loss point, very frequently. So let me just clarify what it is, and then we can move forward. So TLP is basically to-go loss point. So when we are tracking the hydration of any species of the plant, you are concerned with this value. So why is this? Because to-go loss point is the point when the leaves of that particular plant is not upright as they should be when the water is when they are dehydrating, and even in some species after the sugar loss point, even if you give them water, even if you keep them in the light, they don't come back, so the plant gets dried. So this is also directly relevant to the pests as well. So we really try to see the sugar loss point of that particular species that we are working with. So in this case, you can see we have water content on x-axis, and we have peak to peak amplitude on y-axis. What we really see is that as the water content is decreasing, the peak to peak amplitude is also decreasing, which is a very good sign. And we were expecting this. Why? Because when the leaf is fully hydrated, the internal structure is more homogeneous, and ultrasound passes through it more efficiently. As the leaf dries, there is the internal scattering increases and attenuation rises, causing the amplitude to steadily decrease. What really interesting here is that the TLP is basically at a very high value, so 0.973. So what we have observed with potato leaves is that they start to lose their sugar at a very high value, and which we have verified using the destructive testing as well, which I'm going to show you in the next few slides. So we also record time of flight, and time of flight usually reveals a different type of information. So how fast the wave can travel through the leaf. As tissue dehydrates, mechanical stiffness changes, and the internal structure of the leaf is basically relaxed. These changes influence the wave propagation speed, which we quantify using cross correlation between two consecutive signals. So what is really interesting is time of flight is more sensitive at earlier stage of dehydration than amplitude alone. In our experiment, it produced TLP of 0.956, which is a bit different than what we got in the peak-to-peak amplitude, but indicating that the mechanical changes appear early in the acoustic signature. So these features help us understand not only the hydration, but also the biomechanical responses. So we also process the data for frequency doing spectral amplitudes. This gives us and even reveal us more information. So by taking FFT of the received signal, we observe how different frequency bands attenuates as hydration decrease. High frequencies, especially around 650 kilohertz, are particularly sensitive to microstructural changes caused by the water loss. This actually makes sense. So higher frequencies have shorter wavelengths, so they can so they can interact more strongly with small scale anatomical structures. So what we have observed is that we can have even sugar loss point, which is basically frequency dependent. So for example, at 350 kilohertz, we receive a value of 0.943, and even at 650 kilohertz, we receive a value of 0.964. So we can observe frequency dependent shift in the sugar loss points. These variations basically tell us that we can optimize ultrasonic sensing by selecting the frequency bands, and we really have to be very sure that which species of the plants that we are using is basically that we are working with works well with that particular frequency band. So the transmission coefficient is another feature that we usually process. So it provides a normalized measure of how much ultrasonic energy successfully passes through the leaves. Because it is normalized, it becomes less sensitive to external variations like the positioning of the transducers or minor fluctuations in coupling condition. So this is a calibration-free feature. So across frequency bands, the transmission coefficient consistently correlates with hydration levels, with TLP values typically between 0.937 at 350 kilohertz to 0.965 at 650 kilohertz. But the stability makes transmission coefficient especially suitable for machine learning pipelines or for multi-model models that combine several acoustic features. Now, what we have seen is that we have different 𝗘𝗿𝘆 point values using different ultrasonic features that we have recorded, but we really need to validate this. So to do the validation, we have to compare it with the ground truth, and the ground truth in plant sciences is the pressurized chamber method. It is again destructive, but using that particular equipment, you formulate the pressure volume curves, and you track the 𝗘𝗿𝘆 point of the particular species that you're working with. So we contacted our colleagues in Spanish Research Council, and they came back with these values, and then we just compare the values that we have discovered using ultrasonic features, and we compare it with the ground truth. So as you can see here, the most closest value is 0.973. Again, the other values are very similar to one that we obtained using the PV curves or pressure volume curves, but this is the most suitable. Why it is the most suitable? Because it's computationally cheap. We don't have to do anything. It's just a simple 3-4 line course that can do it. So we are going to stick with peak-to-peak amplitude for our next study. Yeah, that's pretty much where the project is right now. We are successful in tracking the hydration status of the plants. So to conclude everything, so the work demonstrated that 𝗬𝗍𝗄𝐿 ultrasound is a powerful tool for non-contact real-time sensing of plant hydration. Peak-to-peak amplitude is fastest and most reliable real-time feature that we can get from the ultrasound. And spectral features also allow frequency optimized sensing algorithms that we can develop in the future. Currently, the project is basically in this stage that we have successfully tracked the dehydration in the potato leaves, and we are going to move forward with the pest infestation. For that, we really need to develop some deep learning models that we are going to do in the next year, inshallah. And there is also a research call, a call for the funding in which we are going to propose a build, so to build an embedded IoT ultrasonic sensor for real-time monitoring. I'm sorry once again that I wasn't able to attend this online, but in the next slide, you can have my contact details. I will try to stay online. If you have any questions, or you can just send me the questions via email. Here are my contact details if you want to contact me, or if you want to have any questions. I'm also including my phone number if you want to contact me on WhatsApp. So yeah, thank you once again to all the team, all the organizers, Dr. Shariar, and everyone who is in the committee for the invitation. Thank you so much.